Hello friends, in this module we will look at the non-heme protein which is hemerythrin. Hemerythrin, it is a non-heme Fe2 protein which transports oxygen in lower organisms. It is found in marine invertebrates and exists in monomeric, trimeric and octomeric forms. Hemerythrin is colorless but oxyhemerythrin exhibits purple-violet color. It has a dinuclear Fe2 centers. You can see here the structure of the deoxyhemerythrin, and after binding to oxygen it gets converted to the oxyhemerythrin. In deoxyhemerythrin, each subunit has two Fe+2 ions bound to a protein. One Fe2 is bound to 3 histidine nitrogen of the protein and the other Fe2 is bound to 2 nitrogen of histidine residues. Besides this, there are two carboxylate groups of the glutamate and aspartate residues of the protein, which form bridges between the two ion plus 2. The two Fe plus 2 ions are also bridged through a hydroxyl group. So if you look at the coordination around both the Fe's, in one Fe you have 3 nitrogens, 2 oxygens, another oxygen from the hydroxyl. So it is 6, that is hexa coordinate. The other Fe, if you see, there are 2 nitrogens from the histidine residues, 2 oxygens from the bridging carboxylate groups and 1 hydroxyl bridging group. So the other Fe is pentacoordinate in the deoxyhemerythrin. So at the active site, as I have just said, one Fe is hexacoordinate and the other Fe is pentacoordinate. And both the Fe, before the oxygen comes and binds, the Fe are in the plus 2 oxidation state. Now when oxygen comes and binds to the Fe, the oxygen binds on the penta-coordinated ion center. As you can see, so the binding of oxygen takes place at the penta-coordinated Fe center, and now the H ion from the hydroxyl group also forms a bond with the oxygen. In this manner, the oxygen which has been bound now becomes a peroxide form species. So, and both the Fe's will get oxidized to the plus 3 state. So, in the deoxyhemerythrin, both the Fe's are in the plus 2 oxidation state. After binding to the oxygen, the Fe's now become plus 3. So, you can see now this happens because each of the Fe gets oxidized to plus 3 by 1 electron each, which this electron is given to oxygen. So now oxygen, which was initially neutral, now gets converted into a peroxo species. Thus, like I've already mentioned, one Fe2 is hexa-coordinated and the other is penta-coordinated. The two Fe2 ions are in a high spin state. There is a weak antiferromagnetic interaction between the two high-spin Fe plus 2 centers by something called as a super exchange through the bridging hydroxyl groups. If you know a bit of the coordination